Here's what we've just shown. If the 5 assumptions are true, we call them the Hardy-Weinberg assumptions because it's based on the Hardy-Weinberg theorem, or it leads to the Hardy-Weinberg theorem. If those 5 assumptions are true, then no evolution occurs. What's interesting then is that we now have an equilibrium. We have a condition for an equilibrium. If the 5 assumptions hold, then the population won't evolve. We call that a Hardy-Weinberg equilibrium or evolutionary equilibrium. Now, there's also another consequence, and that's this. If any of the Hardy-Weinberg assumptions are violated, then evolution can occur. So that means this: the conditions required for no evolution to occur are restrictive, and that's it. If you violate any of the other 5, then you automatically get evolution. But notice there were 5 different assumptions and only one of them was associated with natural selection. Therefore, natural selection is only one mechanism and there are others. How many others are there? Well, there were 5 assumptions. If you violate any one of them being natural selection, there are 4 others. So that means that there must be other mechanisms of evolution besides simply natural selection. And those other mechanisms are all associated with violations of the Hardy-Weinberg assumption. Okay, so what are they? What are those mechanisms? Well, if we look at this, each violation of Hardy-Weinberg, let's go through it again. What's the violation of the large population size? Well, in that case, if you have a small population size, or even if you have a modest population size, another way of saying this, if statistical fluctuations are not negligible, then genetic drift is the mechanism of evolution. Okay, now, here's the thing. You can have this happen in large populations, even no problem, as long as they have enough time for these things to have an appreciable change in the frequency. But this concept of genetic drift is critical. This is a very, very important aspect of evolution. And in fact, there's still an ongoing debate as to which is more important in evolving systems, natural selection or genetic drift. But genetic drift arises by random chance. It's just randomness that occurs. It's easiest to see when the population is small. All right, there's another one. You have violation of random mating. Okay, now in this case, most biologists say that violations of random matings are a special type of natural selection or Darwinian evolution because Darwin wrote about that aspect in another book. But in this case, I'm going to separate it out because it's a different kind of selection. In this case, selection, it's based on what your potential partners are interested in and what they are keying on. So when individuals are not just mating randomly relative to a certain trait, they're choosing one aspect of the trait versus another, then we call that sexual selection. And again, we can classify this, and your book does classify this as a special case of natural selection. All right, what about this? Violation of no migration. What if individuals are moving into the population or going out of the population? Well, if that's the case, if they're not leaving randomly or the population that is sending individuals in has a different allele frequency, then you get evolution by gene flow. You can also get evolution by mutation pressure. In other words, mutations can be so high that it itself is a non-negligible source of evolutionary change. And that's something that we see in for example, HIV. We've already talked about. HIV evolves very rapidly by mutation pressure simply because the reverse transcriptase enzyme is not very precise. It tends to make mistakes. So those are the 4 that are not natural selection. The last one is natural selection or Darwinian evolution. So those are the mechanisms, and they are a bunch of them. Now, let's go back. And we said, okay, in real populations, how often is it that these 5 assumptions hold? And we said, basically, in reality, most populations, those 5 assumptions are not going to hold. So if they don't hold, that means evolution is possible. And that explains why it is that essentially everywhere we look, we find evolution. In essentially every place we look. Now, occasionally, we find things that are not evolving or not evolving rapidly enough for us to notice it. But evolution is so common. It's the most common thing we see. When we find something that's not evolving, we have evidence for something that's not evolving, that's when we're surprised because that's rare. Okay, how can you determine if something's evolving? Right? Evolution takes a long time. How can you determine if it's evolving? The Hardy-Weinberg is what we use. Because here's what we know. if it's not evolving, it will be in Hardy-Weinberg equilibrium. That's the mathematical reality. It has to be that way. So if it's not in Hardy-Weinberg equilibrium, that means at least one of these mechanisms is pushing it away from that equilibrium, and therefore the population must be evolving. Okay, so now we have our way of approaching the question we started with. Is this population evolving? In other words, does TPA25 confer a benefit, or is it detrimental? If it's beneficial, it should be increasing. If it's detrimental, it should be declining. So that's now our tool. We have the ability now to test that. Let me walk you through the logic. We're asking again, is there evidence that TPA25 is evolving in northern Italy, right? Based on that one sample that we have. Okay, so we know the following. We just showed that if TPA25 were not evolving, then it would be in Hardy-Weinberg equilibrium. If it's in Hardy-Weinberg equilibrium, that tells us then that the frequency of plus plus should be p squared, the frequency of plus minus should be 2pq, and the frequency of minus minus should be q squared. Okay, there we are. Now, next bit of logic. We know from the data that we calculated that p is equal to 0.4848 and q is equal to 0.5152. Right, now you know that, and at this point you should know where that comes from. Again, if you're not sure, go back and look at it again before we continue. All right, so now the next step is simply this. Apply this. Apply this to the data. So we know that 0.4848 is p, so that means the frequency of plus plus should be 0.4848 squared, which should be 0.2350. And this should be 2pq, which should be 0.4996, and q squared should be 0.2654. Okay, so this should actually be the proportion that we see in our actual sample. That should be what we see if it's in Hardy-Weinberg equilibrium. So now the question is this what we see? Okay, now you see what we're doing here? This is what we saw in the Mendelian genetics lab. This is looking at what the expectation is from a given hypothesis, in this case the Hardy-Weinberg equilibrium, and comparing the expectation to the data. Remember how we do that? That was the goodness of fit test. And we can use that chi-square statistic in the goodness of fit test to figure out whether or not we have evidence that it's in Hardy-Weinberg equilibrium or not. Okay? Now we have to be a little bit careful here, and we're going to do this very carefully to make sure we get this right. But this is now what our expected frequencies should be. Okay? So, if those are the expected frequencies, what are the actual frequencies? Well, remember, there were a total of 263 people. Now, we can't compare numbers of people to proportions, so we have to convert those proportions to numbers of people expected. Okay, well, that means this. We were expecting 23.5% to be plus plus if it's in Hardy-Weinberg equilibrium. There were 263 people in the sample, so that means 23.5% of 263 is 61.8050. 61.8050 are what if what? Okay, well, 61.8050 individuals, not percent. 61.8050 people in the sample would have been plus plus if it was in Hardy-Weinberg equilibrium, because this is the expectation under the Hardy-Weinberg. And by the same reasoning, 0.4996 proportion of 263 is 131.3948 people. Okay, and then this is 69.8002 people, should be minus minus. Okay, now this is weird. This is saying 61.8 of a person? What does that mean? It doesn't make sense. Okay, well it does. Actually it does. It makes perfect sense. It makes sense in the following way that this statement makes sense. The average number of people in an American household is, number of children, I should say, in an American household is something like 2.4. Okay, average American household is 2.4 kids. Okay, no one on the planet expects to see 4/10 of a kid running around. But it makes sense. The 2.4 still makes sense. What it says is this. Take at random an American household. How many kids does it have? Maybe 3. Take another one. How many kids does it have? Maybe 1. Take another one. How many kids does it have? Maybe 5. Take another one. Maybe 0. Take another one. Maybe 2. Maybe 1. Okay, so take all of those and average them. The average can be a fraction. That's okay. There's nothing wrong with that. Okay, so 2.4 on average. That's what we mean. Think of these numbers the same way. What this is saying is if it's in Hardy-Weinberg equilibrium, there'll be maybe some fluctuations, but the average will be this. In other words, if this population were to go on for eons, and we were to sample 263 people in that population over and over and over again, say 100 million times, then we would get, on average, 61.8050 individuals who are plus plus. In any given generation, we might get 63, or we might get even 52, or we might get even 75. But if we average all of those together, the average number should be 61, which means this: every given year should be pretty close to 61.8050, except for a few oddball years where it was way far out by chance. Okay? That's what these numbers mean. They're on average. It's an on average kind of look at the data. Okay, so let's compare this to the actual data that we got. Okay, well, we know how to do that. Here's the actual data. We got 56 who are plus plus, 143 plus minus, 64 minus minus. That's the observed. Here's the expected. Now, if it's in Hardy-Weinberg equilibrium, we would get close to this. But like I just argued, we won't get exactly that because every year there's a little bit of fluctuation. We've dealt with this before. That's the same thing we saw in the Mendelian genetics lab. So here's the question. 56 is not the same as 61.8, but we're not expecting it to be exactly 61 or 62. It should be somewhere close to that. Well, is this close? Is 56 close? Who decides that? Okay, well, what we have to do now is to decide whether or not we're close enough based on a probability. We don't want to do this by guessing. We want to do this with some sort of objective measure. And that's what the goodness of fit test gives us. Okay, so here's what we're saying. What's the probability? Suppose that our population was in fact in Hardy-Weinberg equilibrium. Suppose that were true. And we went out and we sampled 263 people and we got these results. What's the probability that we would have gotten this big a deviation from these numbers? Notice these are different. What's the probability we would have gotten that big a deviation by chance? Can we calculate that? Yes, that's what the chi-square does. Okay, it gives us a statistic which tells us whether or not the data fit the hypothesis reasonably well or not. Okay, now you know how to calculate it. It's straightforward. We take this number minus this number, square it, and then divide by this number. We get that. Then we take this number minus this number, square it, divide by this number. We get this. This number minus this number, squared, divided by this, and we get this. The chi-square then is this plus this plus this. So we just sum this last column to get the chi-square. And we get a number 2.0522. Okay, now remember, we now have a number that we have to compare to a chi-square critical value. And the critical value is sort of a break point. Because remember, this value of chi-square is telling us how well the data fit the hypothesis. Right? Now, the bigger the number this is, the worse the fit. So, is this a big number or a small number? Well, that depends on the critical value. And again, I have to give you the critical value. But interestingly enough, when you read this in the actual literature, you don't tend to be given the chi-square critical value. What you tend to be given is something called a p-value. The p-value you've read about in the book. We've already talked about it. Here's what it means in this particular case. The p-value is, in essence, the probability you got this big a deviation or bigger by chance, given that it was in Hardy-Weinberg equilibrium. So assuming the population, for the sake of argument, assume it was in Hardy-Weinberg equilibrium and we got this big a difference, we got that by chance, we would get that big a difference or bigger by chance almost 36% of the time. That's what that says. Okay? Now, this then is telling us, well, that's not that unlikely. I mean, if we were to do this randomly, just choose random numbers, 36% of the time it's going to be a bigger difference than this one. So this is telling us that this really isn't all that odd. Another way of looking at it is this. Whenever p is bigger than a number that you have to be given, it's called the alpha number, that alpha number determines what the chi-square critical value is. Whenever p is bigger than that number, the chi-square critical value is smaller. I'm sorry, the chi-square is smaller than the critical value. Okay, so they go opposite. All right, so a large p means a small chi-square. P can only be 1. It can't get bigger than 1. So a p, in this case, that's bigger than 0.05, because I chose, not arbitrarily, but I chose 0.05 as my number, a p bigger than 0.05 means that the chi-square is less than the chi-square critical value, which means it's not significant. So this is telling us the probability that I got this big a difference by chance is not that unlikely. It's a third of the time that would happen. Okay, so there's no evidence here that this is not in Hardy-Weinberg equilibrium. There's no evidence that it's evolving. So we can't say that it's not evolving, however, because we only have a sample of 263 people. So we can't actually conclude from this particular result, although a lot of people do and a lot of professionals do. They inadvertently or mistakenly say that this is telling us that it's not evolving. No, that's not what it's telling us. It's telling us there's no evidence that it is, but there's also no evidence that there isn't. This test can't do that. All this test can do is really look to see if there is evidence for evolution. In this case, there wasn't, but that doesn't mean it's not evolving. It just means this data set doesn't show it. Okay, but now I want you to compare that to the ACE1 data. That was the stuff that I wanted you to compute. Now, I want you to take a moment and compute all of those results. The chi-square value and the, and not the chi-square, sorry, the expected values for the ACE1 under the assumption of Hardy-Weinberg equilibrium. Calculate all that out, get this table, and come back, and then we'll talk about it. Okay, having calculated all that out, these are the expected frequencies or the expected number that of individuals you should get with ACE plus 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 minus 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 if the population's in Hardy-Weinberg equilibrium. You would expect to see 45.5794 who are plus plus, 127.8 who are plus minus, and 90 who are minus minus on average. These are the specific numbers I'm going to use though, because the more decimal places I have, the more precise my answer is going to be. Now, if you go back to the data, this is what they saw. 27 were plus plus, 165 were plus minus, and 71 were minus minus. Now, notice, these numbers are much farther off from the data than these numbers are. So we expect that this is going to give us a bigger chi-square, and in fact it does. If you do all that, you get a chi-square. It's 22 as opposed to 2. So it's 10 times higher, right? Now, we get a p-value then of this. This is, I've got 3 numbers here. What should I look at? The 1.471, the 10, or the minus 5? The exponent. That's the thing you always look at first. Okay, that's 10 to the minus 5, which means I move this decimal place over 5. Okay, there's 4 ones, then the 1. So this is 0.0000149. That's a teeny tiny number. That's way less than 0.05. When this p-value is less than 0.05, that means the chi-square is bigger than chi-square crit. So remember, they go opposite. If p is big, chi-square is small. If p is small, like in this case, chi-square is big, bigger than the critical value if it's less than 0.05. Now, this then is evidence that it's evolving. This means somehow this ACE1 insertion has something going on, and it's probably natural selection. It's probably either detrimental or beneficial. How do we find out? Well, we have to look carefully. If we look at this, it says, okay, well, 20 less than what we expect have plus plus. So this is potentially indicating that it's detrimental. However, if we actually look at research on this particular gene, we'll notice something interesting. There were 2 interesting studies done by different groups earlier than this study was done, in fact. And this one, Riggett et al., in 1990, found that individuals who are minus minus have a circulating concentration of angiotensin-converting enzyme that's double that of people who are plus plus. So in other words, these people have way more ACE than these people. Let's talk a little bit about what ACE is. Angiotensin-converting enzyme is an enzyme that's produced by the lungs. It's a hormone, and it is involved in controlling blood pressure. Now, having too much ACE may be good, it may be bad. We're not really sure. But what this is showing is that individuals who don't have the ALU insertion there have twice as much of this ACE enzyme than people who are plus plus. So somehow plus is apparently interfering in some way with the production of this enzyme. Is that good or is it bad? It's hard to tell, but this evidence is potentially suggesting something interesting because also this is below what you'd expect, so it's hard to tell. But there's another one too. Hamdi et al. In 2002 discovered something really interesting about individuals who are minus. They have a higher probability of having this condition that causes age-related blindness. It's called macular degeneration. The macula is the center part of the retina that is really involved in high acuity vision. People who are minus are more likely to have this age-related macular degeneration than people who are not minus, either plus or plus minus. So that means this: if you're older, you don't want to have minus. You want this in your gene because you're less likely to go blind. Okay, now, how does that affect natural selection? It's unclear. But this is the kind of thought process that we need. First, what we did is we compared what we observed in the data to what we got from the expectation of the Hardy-Weinberg, and we discovered it was way different than you'd expect. Therefore, one of two things is true. Either this is evolving, or this data set is really strange. We just got a very strange bunch of people by chance, which is unlikely. How unlikely is it? The probability that we got such a strange result is less than 0.00001 or 0.002. So its probability is very much on the side of the fact that it's evolving. Okay, so there's good evidence here that it's evolving. Is it evolving through natural selection? Probably, because we've got two different studies at least that show that it has an effect on phenotype, and a potentially interesting and important effect on phenotype. Okay, is it beneficial to have that ALU insertion in your ACE gene, or is it detrimental? Well, this is the only one that's clear. It's detrimental to not have it. It's better to have it if you want to stay, have your vision as an older person. But we don't know what this means. Is it a good thing to have more ACE, or is it a bad thing to have more ACE? The jury's still out on this, but the point is, this is the kind of thing that we tend to see. Whenever we look to see if a gene is evolving, usually it's evolving. Now, there are plenty of opportunities. It's not uncommon to find that the evidence is not showing evolution. But again, that's one of two possibilities. Either it's not evolving, or the study was not strong enough to see it. It's called power. All right. So that's where we are, and that's how we use this Hardy-Weinberg. Now, we're going to come back to this later on when we look at specific genes and see what's happening in populations of humans over time.